Imagine finding out that the grueling chemotherapy designed to save your life is actually building an impenetrable fortress to protect your cancer.
Yeah.
Like that the very medicine intended to destroy the tumor is instead acting as an accelerant, making it significantly more likely that the disease will return and, well, kill you years down the line.
I mean, it sounds completely counterintuitive to everything we understand about modern oncology. You assume we throw our most toxic weapons at the problem and it either works or it doesn't. You rarely assume the weapon itself becomes the cancer's greatest ally.
Right. But that is the shocking reality we are unpacking today on this deep dive. We are looking at a breakthrough June 2026 paper published in Nature Communications, and is completely upending how we look at and treat the most common form of breast cancer globally.
Yeah, ER-positive, HER2 negative.
Exactly. The researchers basically mapped the exact physical coordinates of immune cells inside these tumors, and their findings are poised to spare thousands of women from the brutal side effects of unnecessary chemotherapy.
It is a massive paradigm shift. For decades, I mean, the entire field has operated under a fundamental assumption about the biology of ER-positive, HER2 negative breast cancer. We just assumed these tumors were universally cold.
Okay, let's define that really quick, because the whole hot versus cold tumor concept is super central to this.
Right, right.
A hot tumor is one that the body's immune system has recognized as a threat, right? It's like a loud, chaotic party swarming with immune cells. The cops basically actively trying to shut it down and attack the cancer.
Well, yeah, exactly.
But a cold tumor is flying under the radar. It's a ghost town. The immune system hasn't really noticed it, so there's no cellular battle taking place at all.
And because we assumed this specific breast cancer subtype was, always a cold ghost town, oncologists historically didn't bother looking at the immune environment to make treatment decisions.
They just ignored it.
Pretty much. Instead, they relied heavily on a genomic test called the Oncotype DX recurrence score to predict a patient's risk of late relapse.
Which, I've seen how this test works in practice. It analyzes the genes of the tumor and gives you a score from 0 to 100.
Yep.
And if you get a low score, somewhere between 0 and 15, your risk of the cancer coming back is low. You get prescribed daily hormone therapy, and you can safely skip chemotherapy altogether.
On the flip side, if you get a high score, anything from 26 to 100, your risk of recurrence is severe. In that scenario, hormone therapy isn't enough, and chemotherapy is an absolute necessity.
So the extremes of the spectrum make sense. You have clear binary directions.
Right.
But okay, let's unpack this. What happens to the thousands of patients who get an intermediate recurrence score, like falling right between 16 and 25? Because it seems like the genomic test is essentially throwing its hands up and saying, well, we have no idea.
Yeah, that intermediate group is one of the most frustrating gray areas in modern medicine. The genomics provide no clear answer. And remember, because these tumors were thought to be immune cold, doctors aren't looking at the patient's immune cells for a tiebreaker.
Wow. So how does an oncologist, sitting right across from a patient with a score of, say, 20, decide whether to subject her to hair loss, severe nausea, and long-term cardiac toxicity?
By relying on a shockingly blunt instrument, her menopausal status.
Wait, really?
Yes. The current clinical guidelines recommend that if a woman in this gray area is premenopausal, she generally gets chemotherapy. If she is postmenopausal, she generally doesn't.
That is just a demographic guess. They are using age and ovarian function as a proxy for how aggressive a tumor might be, rather than looking at the actual biological reality of the cancer growing inside her.
It is totally a statistical gamble, and the researchers behind our source material realized that, well, to stop guessing, they needed to start relying purely on pulverized genomics and actually look at the physical architecture of the tumor.
Right.
They needed to see if the cold tumor assumption was actually true in the first place.
Which means we need to talk about spatial transcriptomics and spatial proteomics.
Yes, the heavy hitters.
So the old way of sequencing a tumor is like taking an entire city block, the buildings, the roads, the pedestrians, the cars, and dropping it all into a massive industrial blender.
That's a great way to put it.
Right. And you test the resulting smoothie and say, okay, there are 10,000 cars in this city. You know what is there, but you have absolutely zero context.
You have no idea if those cars are neatly parked in garages or if they are piled up in a catastrophic traffic jam on the highway. Context is everything in biology.
But spatialomics is like firing up Google Earth for the tumor. You take an intact slice of the tissue, place it on a slide coated with millions of microscopic, spatially coded barcodes, and when the cellular RNA binds to the slide, you can map every single gene expression back to its exact physical coordinate.
Yeah, you aren't blending anything.
Exactly. You are creating a high-definition geographical map of the cancer.
And in this study, the researchers applied that Google Earth technology to tissues from 577 patients who were part of a massive, famous clinical trial called TAILOR-X.
That's a huge data set for spatial omics.
It is. And they meticulously mapped out 2 distinct neighborhoods within every single tumor. First, you have the epithelia, which they labeled the EPI. These are the actual nests where the malignant cancer cells live.
The fortified houses where the bad guys are hiding?
Yes, exactly. The second neighborhood is the stroma, or the tumor microenvironment, labeled TME.
Right.
This is everything surrounding the houses, the streets, the structural scaffolding, the blood vessels, the connective tissue.
Okay, got it.
By keeping these 2 neighborhoods completely distinct on their maps, they shattered the foundational myth of this disease.
Meaning the ER-positive, HER2 negative subtype is not a monolith.
Not at all.
When they looked at the maps, they found 4 distinct biological states for the stroma neighborhood and 3 distinct states for the epithelial houses. And while a chunk of these tumors were indeed the classic cold ghost towns, a massive percentage were something else entirely.
Yeah, nearly 30% of the stroma neighborhoods and 28% of the epithelial nests were categorized as immune hot. They were absolutely packed with CD8+ plus cytotoxic T cells.
The killer T cell.
Right, which are the immune system's apex predators designed to hunt down and kill cancer cells.
Okay, here's where it gets really interesting for me. If we stop right there, the logical deduction is that finding a tumor swarming with killer T cells should be an incredible win for the patient.
You would think so.
Yeah. I mean, it means the body has recognized the cancer, the alarms have been tripped, and the immune system is staging an assault. You'd think we should rush in with chemotherapy to help those T cells finish the job.
That is exactly what you would assume. But the spatial data revealed a brilliant, terrifying paradox. The T cells are indeed swarming the stroma, but they aren't fighting. They're trapped in a biological hostage situation.
Wow. Because they can't physically reach the cancer cells.
Right, or chemically reach them for that matter. The researchers looked at the gene expression specifically within these hot stroma neighborhoods, and they found intense tissue remodeling.
What does that mean exactly?
Well, yeah. It means the tumor isn't just sitting there. It is actively weaponizing the body's own wound healing process to build a dense, fibrotic matrix of collagen. It is quite literally pouring concrete walls between the stroma streets and the epithelial houses.
So the immune cells are physically trapped in a maze of scar tissue outside the fortress.
Exactly.
But you mentioned they are chemically trapped, too. The data showed massive upregulation of things like CTLA4 and TGET.
Yeah, those are what we call checkpoint receptors.
Okay.
Under normal circumstances, your immune system has natural brake pedals like CTLA4 and TGT to stop T cells from going rogue and attacking your healthy organs.
So you don't get an autoimmune disease.
Right. But in these hot tumors, the cancer has essentially hacked those brake pedals.
Okay.
It is stepping on them with full force, chemically shutting the T cells down.
And that explains the high levels of CXCL13 they found. It's an exhaustion marker. The T cells are chemically burned out, their radios are jammed, and they are stuck in a concrete maze.
It's a bleak picture.
It really is. But who is doing the jamming? Like, who is pouring the concrete?
The spatial maps pinpointed the culprits. It's a very specific subpopulation of macrophages known as SPP1 plus macrophages.
Usually, macrophages are the garbage collectors of the immune system.
Right.
They roam around eating cellular debris and dead tissue. But in this microenvironment, they are acting as the architects of the fortress.
That is the perfect way to describe them.
Yeah.
These STP1 plus macrophages are actively promoting the dense scar tissue formation, and they are the ones broadcasting the chemical signals that trigger the T cell brake pedals.
They're like bouncers.
Exactly like bouncers. So you have a high density of CD8 plus T cells on the map, but they are exhausted, completely dysfunctional, and actively suppressed by the surrounding macrophages.
Man, so let's bring this back to the clinic. You have a patient sitting in the oncologist's office. She has an intermediate recurrence score of 21. Her doctor's about to flip the menopausal coin to decide if she gets chemo. But if that doctor could pull up this spatial map and see a high density of these stromal CD8 plus T cells, these exhausted, trapped immune cells, does that mean she needs the chemo to blow up the concrete maze?
This is where the paper drops an absolute bombshell.
Yeah.
Having a high density of these stromal CD8 plus T cells, which they abbreviate as STIL CD8 plus, is actually a strong negative predictive marker for chemotherapy in this intermediate group.
Wait, meaning the chemotherapy actively harms their chances of survival?
Yes. The researchers tracked these patients over a 15-year period. If a patient with a high density of these exhausted T cells received only standard hormone therapy, her 15-year survival rate was an excellent 91.3%.
Wow, 91.3%. But what if she received hormone therapy plus chemotherapy?
Her 15-year survival plummeted to just 68%.
That is a 23% drop in survival because of the treatment designed to cure her.
It's staggering.
I mean, I can understand chemotherapy failing to help in a tough microenvironment or causing collateral damage by wiping out the few functional T cells left. But a 23% plunge means the chemotherapy is actively helping the cancer survive and relapse. How is that mechanically possible?
It all comes back to the SPP1 plus macrophages and that wound healing response we talked about.
Yeah.
Cytotoxic chemotherapy works by inducing massive cellular and DNA damage to fast-growing cells.
Right.
But when you introduce that kind of systemic damage into a microenvironment that is already hyperactive in its wound healing pathways, you are literally pouring gasoline on a fire.
Because the body's natural response to tissue damage is to heal it. So the chemo damages the surrounding stroma, and the tumor uses that as an excuse to kick the SPP1 plus macrophages into overdrive.
Exactly. The tissue damage from the chemotherapy likely acts as an accelerant. It triggers the release of alarmins and inflammatory cytokines that amplify the fibrotic remodeling.
It's making the wall thicker.
Much thicker. Instead of clearing the residual cancer, the chemotherapy provokes the stroma into building an even more impenetrable concrete wall. It deepens the exhaustion of the immune system and effectively shields the surviving cancer cells, allowing them to mutate and eventually cause a deadly relapse years later.
We are quite literally watching the weapon fortify the enemy.
Yeah.
This completely invalidates the current standard of care for these intermediate patients, doesn't it? If giving chemo to a patient with this specific spatial map drops their survival rate to 68%, the menopausal coin toss is actively harming women.
It is. And the researchers ran the numbers on what would happen if clinics replaced that menopausal guideline with this new spatial biomarker.
What did they find?
If oncologists looked at the density of these exhausted CD8 plus T cells, instead of asking about ovarian function, it would change the treatment plan for up to 50% of all patients in that intermediate gray area.
That is half the patient population getting their protocols completely flipped.
Huge numbers.
Let's break that down, actually. So what does this all mean for the listener or someone they know trying to make a treatment decision? You have premenopausal women who today are almost universally told they must endure chemo.
Right, and under this new paradigm, 49% of those premenopausal women could safely skip it.
Wow.
They could avoid the toxicities, the early onset of chemically induced menopause, the long-term heart risks, all of it, because their spatial map proves they have the exact kind of exhausted immune environment where chemotherapy will only accelerate the tumor's defenses.
That is incredibly liberating news. But it swings both ways, doesn't it? What about the postmenopausal women who are currently told they are safe to skip chemo based purely on their age?
Yeah, that is the more sobering realization. Up to 50% of postmenopausal women in this intermediate group might actually need their treatment escalated.
Because they're being told they're safe when they aren't.
Exactly. If their spatial map shows they don't have this exhausted, suppressive fortress-like, if their tumor is truly a cold ghost town without the heavy macrophage jamming, they might benefit immensely from the chemotherapy that is currently being withheld from them.
So the current guidelines are getting it backward half the time. And I imagine the researchers didn't just throw this theory out there based on a few slides, right? They had to prove this statistical model actually works in a real-world clinical setting.
Oh, absolutely. They rigorously validated it using something called decision curve analysis, or DCA.
Which is what exactly?
Well, this isn't just looking at basic statistical significance. DCA is a mathematical model that evaluates the clinical utility of a diagnostic tool. It weighs the net benefit of a treatment against the harms of false positives and false negatives across different threshold probabilities.
Okay, so it mathematically proves whether using this spatial map actually results in better human outcomes than the current guidelines.
Yes, and the results were definitive. Over a 15-year horizon, using the CD8 plus spatial marker yielded a significantly higher net benefit than guessing based on menopause.
It actually worked.
It resulted in managing far more patients correctly, sparing the ones who would be harmed by chemo, while accurately identifying those who desperately need it.
It is incredible how far we've zoomed in on this. We started with the assumption of a universally cold tumor. Then we used genomics, blending the whole thing up, only to find a muddy gray area that forced doctors to guess based on age.
Yeah.
But by using spatialomics, by refusing to blend the tissue and instead mapping the precise geographical coordinates of the cancer cells, the immune cells, and the structural stroma, we found the truth.
We found that the location and the context of the cells matter just as much, if not more, than the mere presence of the cells.
Right, because you can have an army of apex predator T cells on the battlefield, but if they are trapped behind a concrete wall of collagen, chemically jammed by corrupt macrophages, throwing a bomb into the mix just gives the enemy more rubble to build their fortress. We are moving from a demographic guess to high-definition spatial biology.
Which is going to redefine precision medicine entirely. It's not just about what genes are mutated anymore, It's about the ecology of the tumor microenvironment.
And that leads to a massive forward-looking question for us to mull over. We just established that if you have this specific immune-hot but exhausted tumor, traditional cytotoxic chemotherapy is the wrong tool for the job. It accelerates the wound healing and makes things worse.
Correct.
But the immune cells are there. The T cells have gathered at the gates. They are just chemically asleep with their brake pedals pressed to the floor.
The infrastructure for an immune response is completely localized and ready.
So if we know traditional chemo is out, could this exact same spatial map be the ultimate key to identifying which early-stage breast cancer patients should be diverted straight to cutting-edge immunotherapies? Like drugs that are specifically engineered to block CTLA-4 and TIGIT, taking the foot off the brake pedal and weakening those T cells up to tear down the fortress.
That is the exact frontier this research opens up. And honestly, it is where the next generation of clinical trials must immediately focus. Because if we can map the exhaustion, we can target the awakening.
It completely changes everything. Thank you for taking this deep dive with us today. For everyone listening, keep questioning the consensus, keep looking beyond the surface, and always pay attention to how the smallest details are shaping our world. We'll catch you on the next one.